Welcome students. This is your third semester course, Technological Methods in Forensic Science. It's quite an important paper. This is an introductory video of what are the different units in that paper, what to expect in this paper, what you're going to learn in this paper. So it's a significant paper where you're going to learn the different technological methods which we use in our science. So it's going to matter in your other senior semesters also, where you're going to learn different specializations where these technologies will be repeated again. So it's an important paper. Okay, so it's a 5-unit paper. I'll just go through some of the important highlights of these units so you get to know what to expect. The first unit is going to be one easy topic, microscopy. Okay, so we all know microscopes. We've all used microscopes. Most of us would have used microscopes as well. So what are these microscopes doing in our science? Okay, so when I say microscopes, they are optical instruments. They're going to use light and their interaction with lenses, for example. So supposing light beams hit a particular convex lens, we know that it focuses in a focal point and the image is magnified multiple times. So we're able to see a bigger image. We also know that we use a certain simple microscope very often in our forensic science, which is the hand lens that we use, simple microscope. So we carry this hand lens everywhere. We are able to see documents. We are able to see fingerprints using these hand lenses. It's an easy tool. Make it a little more complex. We have our simple compound microscopes. So why is this called compound? It's called compound because it has a lens here. It has multiple objective lenses which we can choose. It's also having a stage where we place our specimen, and it finally has a stand. Light passes through here, hits the object, transmits through whichever objective lens we're going to use, then goes to the eyepiece, and we're able to see it here. A magnified image of whatever is placed here in the stage. It's a set of optical lenses, one, and we also have a lens 2. This is a simple compound microscope. But then we all know that in forensic science, not everything can fit into a slide. Sometimes it can be evidence which is very tiny, or sometimes evidence which is a little bigger also. So we have multiple modifications of this compound microscope. We have fluorescence microscope. We have Polaroid microscopes. We also have comparison microscopes, which makes use of a similar setup like we saw in the optical microscope eyepiece. And it has 2 sets of optical objective lenses, which is connected by what is known as optical bridge, 2 stages where we can place 2 objects at the same time and visualize at the same time. We can compare in real time between these 2 objects. That's also possible. Now, this may also not suit our interests at some time. Why? Because the magnification may not be sufficient. Light has its own limitations. So we can use a beam of electrons. So we have what is known as a electron microscope. Okay, so we have electron microscopes which use electrons, a beam of electrons instead of light. So electron microscopes are quite big. So they have what is known as electron source, which hits your sample. And based on the beam of electrons that hits the sample and based on the sample's nature, there are multiple type of effects, these effects are understood and a system is able to identify what is happening. Or this is a one type of an electron, another type of a electron microscope can be a similar setup, a beam of electron hitting a sample, and what passes through helps us to understand what is happening. This is a scanning electron microscope, the first one, and this one is a scanning electron microscope. This one is a transmission electron microscope, based on how they work. Now, when we understand electron microscopes, now that we've understood up to electron microscopes, we can also make it into, have a little more complex microscopes, which we'll be studying, relating to comparison microscopes and what are their applications and things like that. So that will be your first unit. Next off, we're going to have, chromatography. Okay, so what is chromatography? Like the name suggests, it's going to be, something with relation to colors, the chroma that is there. So we're going to use different mediums. Okay, so say, take for example a simple sheet of paper. We're going to run, for example, plant pigments. Okay, take a leaf, extract from the leaf, spot it on this paper, allow alcohol to run through the paper. So alcohol, because of your capillary pressure, capillary force, alcohol is going to rise up through this paper. While it rises through this paper, it's also going to carry these plant pigments that we spotted, and finally we're going to get a paper with multiple bands. So we could get a green band here, we could get a blue band here, okay, and supposing we also get a red band here. Okay, so based on the different bands that are formed, we can identify which plant maybe that pigment is from. We can also use it for inks tomorrow, to understand what type of ink was used to make that document, a question document perhaps. Substitute that paper into silica gel. Now spot your samples, have a sample jar where you're going to have the solvent mixture, like your alcohol that you added here. Supposing you're going to add a mixture of organic substances, that is going to rise through the paper vertically because of the capillary force, capillary action, and the sample is also going to be pulled up. Okay, and now you have again multiple bands. Based on the multiple bands, you're going to identify which sample your test sample is actually consisting of. That is possible. Now, if you want to stop talking about liquids and you want a little more advanced chromatographic technique, you have gas chromatographs, which work on gas mixture. So a person who is drunken driving, who's caught on drunken driving, he may have an alcohol in his system, ethanol, for example. In this case, we put it through a column which is coated with silica, just like what we saw in the previous example, and it's going to elute out at different times from this silica column. If you're going to get an ethanol fraction, it means that he is drunken. If there is no ethanol fraction, he's not drunken. This is a simple application of chromatography. We'll be learning about multiple types of chromatography in our classes in Unit 2. Unit 3 talks of colorimetry and spectrophotometry. So here we're going to talk about different interaction of light with colored objects. You may have learned it in your schooling also. So supposing you have a blue solution, okay, a blue solution of say copper sulfate, Simple example, you're going to pass, red light over it. Red light gets absorbed by this copper sulfate solution, and you're passing a lot of red light. Very little red light comes out because a lot of red light gets absorbed by this solution. Based on this absorption here, you are able to identify the concentration of this copper sulfate. This is the basis of colorimetry. So we're going to study about colorimetry techniques. We can use natural light, we can use ultraviolet light. Okay, so spectrometry and spectroscopy, what happens? We are going to use different forms of light. So supposing you are having UV light or you're having infrared light. Okay, so you want to know what your sample is. It can be a gemstone, for example, or it can be some other sample of a solution, perhaps. Based on the different interactions of UV light, based on the different interactions of IR light, what is the output going to be? Depending on that output, you will be able to understand what these objects are. That is the basis of spectrophotometry. So we have UV spectrophotometry, you have infrared spectroscopy, you have multiple types of spectroscopy based on what you are using. So it's not only ultraviolet light and infrared light, you can also tomorrow use a beam of electrons, you can use multiple types or atomic waves. Okay, so there are multiple things that you can interact your sample with and understand their nature. That is your unit 3. The 4th unit, we'll be learning electrophoresis and neutron activation. Okay, so you must have come across this term again, electrophoresis. So we're going to use electricity, okay, in order to bring our interaction in a faster pace. So supposing you have a gel plate, you have antigen here, you have a antibody here, I'll code it with red. You keep it for 24 hours or so, there is migration. And if this is the specific antigen and antibody, you're going to get a band here. You need 24 hours perhaps to do that. But in case you're going to use some amount of electricity now, you have antigen here, antibody here, and you're going to connect your plate to electricity now, okay, to a battery source, and you're going to run the reaction. It's going to happen faster. You're going to get a band within minutes. So you can use electrophoresis to study antigens, to study antibodies, to study DNA bands, fragments of DNA, multiple polar things. So we'll study the different types of electrophoresis that we will be covering in our syllabus. Next off, neutron activation. So supposing you have a trace amount of metal in a, say for example, it's a case of firearm injury, you have a small bit of bullet piece or cartridge piece, which you need to identify whether it's made of a particular alloy. You don't want to waste it. The best way to identify very finely, very precisely what that bit of metal contains is to ideally use neutron activation. So we bombard it with neutrons, we activate it, we activate that metal, it starts producing gamma rays. Measuring these gamma rays, we will be able to identify what this metal is. We will be learning about neutron activation analysis and its forensic significance in our 4th unit. Now the last 5th unit will be photography. So we all know what photography stands for. Okay, so we have a camera, we have a lens in the front, we have a picture in the front, sorry, and a lens inside, and then a screen which captures the image. So you have an image of a tree, It goes through the aperture, hits the lens, and a image of the tree is formed here. So we used to have optical cameras before. Now we have digital cameras which have better sensors here. So we have CCD sensors, CMOS sensors. We have multiple ways of sensing the object and forming an image. We also have videography using motion pictures. Supposing a person is running, okay, from one point to another, we capture using a same setup, but we're capturing a motion picture of the person running. And now you have a videographed video, a clip of that moving person. Okay, so these are common ways in which we take photographs and videos. But how is it important in a forensic scenario? We take crime scene photographs, we take photographs in the laboratory of our findings, we take videographies of crime scenes. So we need to understand the different concepts in photography. We have photography which involves today even UV photography, IR photography, which can get you best clarity pictures. You have 3D photography, which is coming up. So there are multiple types of photography that we are slowly starting to venture into, and it's important for us to know some of them. So this paper will be studying these 5 units. I hope you at least got an idea of how it's going to go from here. Okay, so we'll be covering one by one.